All right, thank you very much. Hello, my name is Laura Balter. I am today presenting to you the contents of my still ongoing master thesis on land-based cultivation conditions and the enhancement of antioxidant contents in the green macroalgal genus Umbraulva. My thesis is part of a larger project called Mak-Pak Scale-Up. This is a collaboration of several research and commercial partners that aim to produce a sustainable, compostable, and ideally also edible food packaging from green macroalgae belonging to the family of Ulvaceae. On the right here, you can see one very prominent member, the sea lettuce or Ulva lactuca. We also hope to prolong the shelf life of the packaged food and provide some health benefits for the consumers by increasing the antioxidant contents of the algae during the cultivation. The working group at the Alfred Wegener Institute that I was part of concentrates on researching methods of reproduction and upscale cultivation that can then be applied by our agricultural partner, Rovial. My thesis is located within the step of upscale cultivation. That's what I will be looking into. Our other research partner, Hochschule Bremerhaven, is looking into methods of processing that biomass into the product, which will then be implemented by the commercial partners Hengstenberg and Fasergusswerk Polenz. And the finalized product will then be marketed and sold by the German fast food company Nordsee. In my thesis, I look at the species or a species from the genus Umbraulva. The specimen that I was working with were kindly provided by the Nordsee Aquarium in Bremerhaven. And since these originated at the North Sea island Helgoland, I presume that they belong to the species Umbraulva dangiardi, as that is the only one that has been identified there. The entire genus is characterized by the thin and soft dystromatic thallus, so a thallus that only consists of 2 cell layers. And as you can see on the picture on the right, it has a very characteristic olive green color, which is caused by the yellowish pigment called siphonoxanthine. This pigment helps the organism to utilize even green light for photosynthesis and is therefore an adaptation to the shallow subtidal habitats that it inhabits, compared to the more intertidal species of Ulva who do not have that pigment. Other than the pigmentation and the taxonomy, this genus is very barely studied, so it is a very interesting organism to look at. My thesis overall can be divided into 2 topics. The first one are the general long-term cultivation conditions. There are 2 main experiments that I did here. The first one looked into different types of seawater. I used as treatments natural seawater from Helgoland, artificial seawater used in the research facility of the aquaculture at the Alfred Wegener Institute, and artificial seawater that had already circulated through the exhibition aquaria at the North Sea Aquarium in Bremerhaven, so reused artificial seawater. In the second experiment, I also wanted to look at the size limitations that of the setup that I was using. So I compared 1-liter vessels with 5-liter vessels. Here I expected that since both the natural seawater and the algae originated on Helgoland, that this would be the most beneficial for growth of the algae. And based on a study by Oka et al. In 2019, I also expected that the 1-liter cultivation vessels would actually result in a higher growth. This is due to the fact that species from the family of Ulvaceae are known to need a balance between enough space to grow and also enough self-shading as a protection from light damage. Speaking of light damage, the second part of my thesis looked into settings that could be applied right before harvesting the algae in order to enhance the antioxidant contents. As antioxidants are a countermeasure against light stress, I played around with light settings. In the first experiment, I compared the normal white cultivation light with a blue light setting. And in the second experiment, I compared the cultivation setting of 90 micromole photons per square meter in second during a 16-hour day with a higher light intensity of 240 micromole photons per square meter in second. I maintained the same overall daily photodose of 5.18 mole photons per square meter by reducing the day length to 6 hours. And in the final treatment, I kept the high light intensity of 240 micromole photons, but also but let that run for a 16-hour day, increasing the overall total daily photodose. I expected that, based on previous studies, that the blue light, as well as the high intensity and the high daily photodoses, would increase the antioxidant content of my algae. These hypotheses were tested in the following setup. The similar setup was used for all of the experiments. So I had 6 replicates per treatment. Each of the replicates held discs with a diameter of 2.4 centimeter that were cut from the Umbraulva thallus. And the algae were kept at 15 degrees Celsius with water being exchanged and nutrients added every 4 days. On the right here, you can see the setup of the vessel size experiment with the smaller and the larger cultivation vessels. On the top right here, you can see how I cut the discs from the thallus with a cork borer. And at the beginning, end, and during the experiment, several variables were measured. First, of the size. This was done by taking pictures of the laid out thalluses and thalli and an analysis in ImageJ of those pictures. I also calculated various variables, including the relative growth rate and the stocking density. I furthermore measured the photosynthetic activity in FV over FM, as this has been a widely applied measure of overall well-being for algae and plants. For the light experiments, I also did an ABTS radical cation decolorization assay in order to quantify the antioxidant contents. My statistical analysis was conducted on R. I first checked the normal distribution and homoscedasticity of my data. Then I ran a repeated measures two-way ANOVA to compare the effects of the treatment and the duration of the experiment on the measured variables. Repeated measures here, as I used the same replicates throughout the experiment. I changed that approach afterwards so that I could do conventional two-way ANOVA for the vessel size and the light quality. And apart from the initial measurements, I only did one more measurement for the light quantity experiment. I did a one-way ANOVA here. And in cases where a significant effect of either of the factors was detected, I also did a post-hoc pairwise comparison using 2pHSD. So let's look at the results. For the seawater type experiment, we can see that the overall photosynthetic activity was relatively stable throughout the entire 2-week experiment. The reused artificial seawater performed lowest, but all of the values were between 0.6 and 0.75. Overall, I was able to see an increase in surface area for all of the treatments, with discs in the natural seawater growing largest. Both the natural seawater and the artificial seawater actually caused a stagnation in surface area after day 11. This also corresponds with what we see in the relative growth rate. We started out with a relative growth rate of 14 to 20% increase per day, which then steadily declined towards 0 towards the end, with the reused artificial seawater having the lowest decline. For the vessel size experiment, just to give you an idea, this is the difference between the stocking densities in the two vessels. So the 1 liter vessels obviously had a much more dense population. And then I was only able to record a significant difference between the two treatments on day 14. So here, as expected, the discs in the 1 liter beakers did grow a bit larger. And for the relative growth rate, there was, again, the same development as in the first experiment, with no effect of the treatment on the variable. Moving on to the antioxidant enhancement experiments for the light quality, we had a slight problem. I initially intended to run the experiment for 7 days, but on day 6 we had a malfunction of our culturing room, which increased the temperature temporarily to 23 degrees, so I was not able to use that data. But even at 4 days, we can see a difference in the photosynthetic activity uninfluenced by the treatment. And the values, again, are still between 0.6 and 0.75. And interestingly, there was an increase in antioxidant content, but in the white light setting, not in the blue light setting as expected. And for the light quantity experiment, I was able to see that so here you can see on the left the initial measurements, and then the 3 other treatments were measured after 4 days. So the control setting with 90 micromole photons per square meter in second remained the same in terms of photosynthetic activity, whereas for the high light intensity settings, the photosynthetic activity sank far below 0.5 in both settings. This was, however, not translated into a production of antioxidant content. I was not able to see any difference between the treatments here. Those were a lot of statistics, so what do they actually mean? For the first experiment, I was able to show that the natural seawater did promote the best growth, simply, in my opinion, because both the Ulva and the seawater stemmed from Helgoland, and it is very hard to simulate natural conditions with artificial seawater, even if it had gone through the exhibition tanks. The initial relative growth rate that I recorded with 14 to 20% increase per day was relatively high compared to other Ulvaceae. And there was a decline over time, which is very natural simply due to nutrient limitations, or more likely space limitations. This space limitation was, however, not confirmed in my follow-up experiment, as the 1 liter vessels actually proved to be quite sufficient. There is a study by Oka et al. That has provided the threshold value of 187 gram dry weight per square meter as the optimal stocking density for an Ulva species. So it would be one further step to kind of try to convert my surface area values into that and compare it. And overall, it can be said that the algae remained very within healthy levels for green algae, so between 0.5 and 0.8. For the enhancement of antioxidants, I was not able to produce the expected enhancement by blue light. This is probably due to the fact that Umbraulva is a rather subtidal species, so it is more used to blue light than an Ulva species that lives in the intertidal range.
2 more minutes.
Yeah, thank you. And there was no effect of the higher light intensities or higher photodose, and this does not, so this can either mean there was no antioxidant increase, but it can also mean that I just simply did not detect it due to the chosen measurement range or the chosen measurement method. I want to add here that I also recorded further variables that need to be analyzed, such as the specific growth rate, the doubling time, and also for the vessel size and the light quantity, I measured weight. Dry weight of the samples. I also did a nutrient uptake experiment to ensure that no nutrient limitation occurred during my experiments. So in conclusion, my first hypothesis was confirmed by my experiments. I was able to show that the natural seawater and the stocking density in the 1 liter vessels were quite beneficial, and these settings can be recommended to our agricultural partner. For my second hypothesis, I was sadly not able to confirm this, so more experiments need to be run to get a conclusive answer here. I would suggest experiments that include a shorter measurement period in terms of hours or days, other light variations, so light that might not reach the subtidal habitats like red light, infrared light, UV light, and also to combine different methods of antioxidant quantification and determination methods in order to get the whole picture. My personal next steps will be the in-depth analysis of all these variables and the writing and submission of my thesis manuscript in early August. With that, I would like to thank you very much for your attention. I would also like to thank Laurie Hoffman and Bela Book for their supervision. I would also like to thank Inka Bartsch and the working group of Rocky Shore Ecology at the Alfred Wegener Institute for providing the lab space and the technical support, and Dr. Heike Kück at the Nordsee Aquarium Bremerhaven for her cooperation and the provision of the specimen that I was working with. Thank you very much, and I am curious to hear your questions now.